Good afternoon, everyone. My name is Rudolf Klaver. I'm a CEO of Coave Therapeutics. So Coave is a genetic medicine company based on a unique delivery platform. We have an initial focus on CNS and ophthalmology disorders. So what we want to achieve at Coave is really to develop the next generation of vectors that will enable to move from ultra-rare disease to more prevalent disease, and to do so, what we have to do is to really find vectors that will be efficient to target extrahepatic tissues and to be able to deliver not only a gene-for-gene replacement or silencing, but also vectorized biologics or gene editing. So our technology is pretty unique compared to the competition. I will come back on that in the next slides. But basically what we do is we start from existing and established vectors such as AAVs or LNPs. We don't modify those vectors genetically. What we do is we use a unique conjugation technology to conjugate rationally designed ligands to the surface of AAV. So our core technology is a linker technology, which have been developed and very different from what is used for ADC, for instance, which enable in one-step chemistry without catalyst to bind covalently ligands to the surface of AAV, and we can achieve up to 500 ligands on the surface. We have scaled up that technology up to GMP-like conditions, and we have demonstrated the reproducibility and the high yield of coverage and of conjugations and recovery of AAVs in this technology. It's fully scalable and transferable. The objective of redecorating those AAVs really to improve two key aspects: the efficacy in both biodistribution and transduction, but as well improve the safety profile of these vectors. So what we have demonstrated in multiple NHP studies and rodent studies as well is we can, with different family of ligands, improve the biodistribution of the capsid. Specifically, we have a family of ligands that enable to basically change the interactions of the AAV with their natural receptors that enable really a spreading of the vectors in the extracellular matrix and improve then the targeting of tissues. We have ligands that have been developed specifically for pure tissue targeting and detargeting. So I will come back on this. Those ligands bind to specific receptors and will enable to target only a dedicated tissue. And finally, we have now a set of data showing that the presence of those ligands to the surface of AAV prevents the interactions with the existing neutralizing antibody, but also we have a study showing that we generate less antibody against the backbone capsid. So we are very different from the competition because the principle of our technology is working in a different way. So most of the existing AAV companies are using genetically engineered AAVs, mostly using directed evolution. The principle is they generate billions of capsid randomly. Then they screen those capsid by multiple cycle of enrichment in different animal species and primates until they find a capsid that will have the desired property. And then we have these new variants. They are looking for the mechanism of actions that explain the property of this capsid. So for instance, is this capsid binding to specific receptors? And when they find the receptors, they have to demonstrate these receptors is actually conserved in humans. So that last step would be only validated in clinical trials. So it takes multiple years to generate such a capsid. It takes dozens of animals and a lot of money. What we do, it's actually the other way around. We start from an existing and established mechanism of action. So a receptor that is already known, and we have selected ligands that bind to these receptors, and we focus on receptors that are already expressed in human or shared across species. And then we start by screening the ligands. So we don't screen the capsid first, we screen the ligands, we design the ligands, and when we have selected a handful of best ligands, then we conjugate them on AAV. So what does it mean? It means that basically we are not screening billions of capsid, but few dozens of capsid, and we need only one cycle of screening before finding the lead capsid, and we do an additional validation, and then we have the lead capsid. What we think also is because of this process where we start from the mechanism of actions that are already established, we have a higher rate of or probability of translating these properties in a primate to human. So we have two lines of development. Come back. One line is based on sugar-based ligands, so ligand for which the end group is a different glycan. And those ligands have been developed specifically to improve biodistributions of AAV capsid for local route of administrations. And we have another line of development for peptide-based ligands. I will explain a little bit more for systemic administrations with clear tissue targeting. For the sugar-based ligands, we have developed capsid for brain and retina with different routes of administrations. In all those routes that are mentioned here, we have a demonstration in primate of superiority of our capsid versus the best in class. Currently, we are focusing mostly on intra-CSF capsid and suprachoroidal administration in the eye. Today, I will not show the suprachoroidal data from our recent NHP study because this data will be presented already at the AGCT in a month from now. So for the CSF capsid, the strategy was actually to improve the current standard, which is the AV9. So what is known is when you inject AV9 in an intra-CSF administration, such as in this case ICM administration, you have less than 1% of the capsid going to the brain. It goes to the spine and mostly it's spread to the rest of the body. So you have a very limited targeting. Most of the company using this route on this capsid has to push for a very high dose. One of the key issues as well is the AV9 is targeting some tissues such as dorsal root ganglion that may lead to toxicity. So the idea was really to improve in one hand the targeting of the brain regions, but also to decrease DRG and liver transduction. So we have run a first study in primate. It's a multiplex study where we have tested a dozen of capsid, including AV9 and AV2. AV9 is a comparator, and a conjugated capsid that are conjugated AV2. And what you have here is a presentation of the mRNA, so the transgene expression level, fold change versus AV9. And what you see is those different structures of the brain, from the cortex to even deep structures such as the striatum. We have improved dramatically the level of transduction for a 5-fold to a 100-fold time versus AV9. At the same time, we have improved transductions of those brain area. We have decreased dramatically the transduction of DRG from 30 to 40-fold less, as well as the liver. So we have achieved this desired profile of vectors with improved transduction of the brain, decreased transductions of the DRG and liver. So that was our first studies, multiplex in primate. We have very recently conducted a second study, which is a simplex study. So we are validating the vector, one vector per animal. And this is the data. We are still analyzing data, and we're going to generate soon the molecular biology data. But this is the immunochemistry quantification data, comparing AV9 on the left, so where we have looked at three regions, the primary motor cortex, insular cortex, and putamen, versus our conjugated AV2. And what you see, obviously, is we have improved. This is at the protein level, the protein expression from 70-fold to more than 100-fold in the putamen using our conjugated AV2. Very interestingly, and something we have seen in the previous studies we conducted in primate for intraparenchymal administration, is the conjugations of those sugar ligands to the AV does not change the cell specificity of the original serotype. So AV2 has a very good specificity to neurons as opposed to AV9, which transduce a lot of glial cells, which might lead to inflammations. Here we see again that the conjugated AV2 are mostly transducing neurons. So we have a conjugated AV2 which is better than AV9 in terms of transductions that transduce neurons and is detargeted for DRG. We're going to have more on this data presented in the coming month. So we are now in the phase where we are exploring other moieties and other ligand families. So we started with the small molecule glycan-based ligands. We have now peptide-based ligand, and I will explain and show you some data. We have generated ligands with aptamers, and we are working on new ligands with larger proteins and end groups such as CAV or nanobodies. Again, all those ligands are based on the same technology, which are our linker technology, our chemistry to bind. Recently, we have generated also data showing that we can conjugate LNPs, post-packaging of LNPs using one of our ligands. So technically, we can produce one ligand and conjugate LNPs and AAV with the same ligand. So there's great opportunity here to use that for especially prime editing, where the use of AAV and LNPs should be done concomitantly. We have a strong IP positions around, of course, the conjugations, our technologies, and the product that will use these conjugated AAVs. So talking about the peptide-based ligand, here our approach is different. We are looking for tissue targeting. We are looking at the specificity issue of AAVs for IV administrations with currently 2 lines of development, one for targeting BBB crossing and one for targeting skeletal muscle. We have generated a family of peptide-based ligands that are binding to transferrin receptor 1, which is a well-established actually mechanism for crossing the BBB. We have already generated conjugated AAVs, and we are currently testing in vitro and in vivo our first conjugated AAVs. For the muscle, we are using also transferrin, but also an integrin binding peptide, so a new family of ligands we have developed. So this is an example of what we have generated with peptide-based, so-called RGD peptide binding to specific integrins. On the left-hand side of these slides, you have the conjugation data set. So what you see is the nice shift of the mass of the VP size, and we can play with the conditions to increase the density of ligand conjugated. So we can go from 20 to 200 ligands to the surface of the whole AAV capsid. You have the mass photometry data showing a very nice shift of the whole capsid mass. So it's an AAV2 in orange. You have the 2 peak of the AAV2 preparations prior conjugation. And after conjugation, you see the blue peak is shifting. So we have a very narrow peak, just confirming what we have seen in previous studies with the sugar-based ligands, that we have an homogeneous conjugations of the capsid. On the right-hand side is our first demonstration that the conjugated AAV2 with this integrin binding peptide actually bind the integrin receptors. So we have used a soluble receptors to demonstrate that there's an effective binding to those receptors, and that has been recently shown through dot blot analysis. And we are now moving this capsid to IV administration in vivo to generate data showing that it can bind to the integrin in the desired tissue. So in summary, we have a unique platform that could be used directly on any kind of AAV serotype. We have actually ongoing collaborations with companies that have developed candidates on using a certain AAV serotype, and we can directly modify it without changing the whole capsid. So this is the beauty of this technology. We have so far demonstrated that we can improve the capsid for CNS and eye, but we think we can really deploy that technology to different tissue and organs. We have also an internal development. We have 3 preclinical programs in ocular and CNS space with in vivo POC, including a nice biodistribution study in primate, but for the sake of time, I will not share that with you today. We think one of these programs could reach IND by the end of 2026. As I said, we have also partnership, and we are eager to discuss partnership with companies. We have one partnership with a big pharma and one with a NASDAQ-listed biotech company. Both are evaluating currently our technology in NHP model. And this is it for today.